I think it's very important for me that a student who's done an introductory course in biology should be able to read the primary literature in that field, should be able to read what scientists are doing now and understand it. And so this has always been one of the aims of our course, and this is really one of the aims of the book. We want students to be able to have a foundation so that when they encounter something they're unfamiliar with, that science is unfamiliar with, they can make an understanding of it. And I think we've done that both in terms of the actual text for the book, the ancillary resources and so on, and also by bringing in examples from classic and current research in the book. So students actually see science that is going on. They see figures that are from the primary literature. They solve problems based on things that came up in primary papers. I think that's a really important part of being a scientist, and the whole aim of of the course as well as the book is to turn interested people into scientists.
And so we have in every chapter, almost every chapter, we have a separate box, experimental tools. So, and for example, the chapter on DNA structure, chapter 2 introduces the concept of the base pair, but then we have a series of boxes as how does the base pair lead to these experimental methods. So we've separated so that we have toolboxes, they're called. And then, but what that allowed us to do was to then talk about the intellectual processes that go into the research. How did they think about the problem? How did they understand what the problem was? How did they define it? How did they set up their next set of experiments? And so that we're not just focusing the students' attention on the experimental methods, but actually on the thought process that went into designing the experiments.